Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 3 for October 2022. Let us begin with the first question. Question 1 says, plant foods in the diet contain antioxidants such as vitamin C. Storage time and conditions affect the concentration of vitamin C in these foods. They want you to explain how increasing the quantity of plant foods that contain vitamin C in the diet may protect against heart disease. We know that vitamin C is an antioxidant, and antioxidants reduce free radicals. These prevent damage to tissues as well as endothelial cells of the blood vessels, so atheromers as well as plaques will not be formed, or at least there will be reduction in the formation of these plaques, and that will lead to protection against heart disease. Moving on. Part B says, the effect of storage time and temperature on the vitamin C content of all foods was investigated. They want you to describe a method to allow a quantitative comparison of vitamin C content of two of these foods. So here we have together foods, we squash them and dissolve them in the same volume of distilled water, and then we have to make sure that we measure a specific volume of DCPIP into specific test tubes, and then add the vitamin C solution and see the volume required for the DCPIP solution to turn colorless. So here I said, to the same mass of each of the food sample, add the same volume of distilled water, and then macerate to ensure that vitamin C is extracted as much as possible. Then measure out the same volume of DCPIP in two separate test tubes. For example, it could be 1 centimeter cubed. And then add the food extract, I'm talking about the solution, using a clean syringe and the vitamin C standard of non-concentration drop by drop into the corresponding test tubes while swirling until the DCPIP solution turns color from blue to colorless. Then you will record the volume of each solution that decolorizes the DCPIP solution, and then calculate the concentration of the unknown vitamin C extract. As you can see here, it's going to be the concentration of the standard times the volume of the standard divided by the volume of the vitamin C extract. So we can see this is 1, 2, 3, 4, 5. There are already 5 marks even before these two. So if you had written something close to this, you would get the 5 marks. Down here they say, state the independent variable in this investigation. The independent variable is something that is being changed or varied. In this case, it's the type of food. Moving on. Here they say, the effect of storage time and storage temperature on the concentration of vitamin C in different foods was investigated. The juice from 4 foods were stored at 4 degrees Celsius and 20 degrees Celsius for 4 weeks. The vitamin C concentration was measured each week, and the graphs show the results of the investigation. So we have the orange, we have lime, we have carrots and pineapples, and we can see overall, because this is at 4 degrees and that is at 20 degrees, the lower the storage temperature, at least for oranges and lime, the faster the concentration decreases. But in the case of carrots and pineapples, we can see the one stored at a higher temperature decreases rapidly, so we can see storing at 20 degrees leads to a faster decrease in the vitamin C concentration. So here they say, calculate the rates of change of vitamin C content in oranges for the first 3 weeks of storage at 4 degrees Celsius. They want you to give your answer to 2 significant figures. At 0 weeks, we had 4.4, and then at 3 weeks, we had 1.6. So I'm going to calculate the rate. The rate is going to be 4.4 minus 1.6 divided by 3 minus 0, which is 2.8 divided by 3. And it gave me that, but to convert it to 2 significant figures, I got 0.93 gram per decimeter cube per week, and that's my answer. Then they say comment on the effect of storage time and temperature on the vitamin C content of these juices. So this is a 4-mark question. I went all the way, and again, looking at the graphs, I said as storage time increases, the concentration of vitamin C of all the juices decreases. We can see they all decreased. And then here, there was greater decrease if stored at lower temperature. Here, there was greater decrease if stored at high temperature. So here I said, as storage time increases, the concentration of vitamin C in all the juices decreases. For oranges and limes, storage at 4 degrees Celsius leads to greater decrease in vitamin C concentration than storage at 20 degrees. For pineapples and carrots, storage at 20 degrees leads to a greater decrease in vitamin C concentration than storage at 4 degrees. And again, this is true. You can see at higher temperature, there was a greater decrease than at 4 degrees. Then carrots had the least difference between results at 4 degrees and at 20 degrees. At day 4, limes had the greatest difference in the results at 4 degrees and 20 degrees. And finally, the results had no standard deviation, so validity can be established since it's unclear if overlap occurs at the different temperatures for the same juice. So this was comment. That is why I put a point about the validity or commenting on the validity of the results. So this brings us to the end of question 1. Let's continue to question 2. Question 2. Pineapple plants contain the enzyme bromelain that breaks down proteins such as casein. Name the bond between amino acids that is broken by this enzyme. This is a peptide bond. And next they say, name the process that breaks the bond between amino acids. This is going to be hydrolysis. And next they say that digestion of casein by bromelain was investigated. The amino acids in casein, a milk protein, were stained blue. For concentrations of this casein were used. They say a calorimeter was used to measure the absorbance by the blue amino acids released in this reaction. The more digestion that occurred, the more blue amino acids were released, and the higher the absorbance rating on the calorimeter. The graph shows the results of this investigation. We can see for the different concentrations of casein, as the casein concentration increases, the absorbance increases with time. So here they say the initial rates of reaction can be obtained from this graph. The table shows the initial rates of this reaction. So we have that corresponding to that, and they wanted us to find the value at 3.0. So to find the rate for 3.0 substrate concentration, I had to find the gradient of that line. So which is, I chose that point as well as that point, which are 40, 0.72, and 20, 0.36. So I used them to calculate here, 0.72 minus 0.36, divide by 40 minus 20, and I get 0.018, which is what I wrote here. And down here they say, plot a suitable graph to show the effect of substrate concentration on the initial rate of reaction. Here we have initial rate of reaction and substrate concentration. So as the substrate concentration changes, the initial rate also changes, and we can see the change is in a linear manner. So initial rate of reaction on the vertical axis with its units, and here substrate concentration in percentages. So I chose 1, 2, 2, 3 based on the information we have here, and then I chose a corresponding appropriate scale for the vertical, and this was my graph. So moving on. Here they say calculate how many times faster the initial rate of reaction is for casein concentration 2% than the casein concentration at 0.5%. So we have 0.5% is that, and 2% is that. So we're going to use 0.014 and 0.008. So dividing that by that, we get 1.75, which is about 1.8, because our answer has to be to two significant figures. So it's 1.8 times more. Next they say, explain the shape of the graph you have plotted. Remember, the graph was linear. This one here. It's a linear relationship, and it's a positive correlation. As substrate concentration increases, the initial rate increases. Now, because this is substrate concentration and this is rate of reaction, we have to talk in terms of enzyme substrate complexes being formed. The higher the substrate concentration, the more the collisions between the enzyme and the substrate, the higher the rate of formation of the enzyme substrate complex, the higher the rate of reaction. So I said the graph shows that as the substrate concentration increases, the initial rate increases. The shape is linear. Because increase in substrate concentration increases the number of collisions per volume, so more enzyme substrate complexes can form and the rate increases. Since the graph does not level off, the enzymes are not yet saturated, meaning the enzyme concentration is not limiting the rate of this reaction. Moving on. Here they say, bromelain can be used to prevent apple juice from going brown. Bromelain can be extracted from the stem and the fruit of a pineapple. The effect of pH on these extracts was investigated. The graph shows the result of this investigation. So we see the enzyme concentration on the vertical axis, as well as pH on the horizontal, and we can see for both the optimum pH is about that, at least based on the results we have. And as pH increases, enzyme activity increases more in stems in comparison to the fruit extracts. So here they say draw a suitable table to show the results of this investigation. This is quite easy. At pH 4, we can see that and that value, which is 0.7 and 1.8. Then we have at pH 7, we have that and that. I recorded them 2.3 and 5.1. At pH, we have 8.5. We get that result and that result, which is 2.5 and 6.6. And at pH 10, we have that and that, which is 1.5 and 1.9. In order to get through max here, you have to put the independent variable first here and then the dependent variable on the other side. And then you have to put appropriate labels as well as units. Moving on. So here they say, determine which of these extracts is most useful in preventing apple juice from going brown. Of course, from the experiment, we saw stem extracts are most useful because the enzyme activity was higher than in fruit extract. If you look at the graph, we have the pH 8.5 where we had the highest enzyme activity. So I say that pH 8.5, enzymes in stem extracts are 2.64 times more active than those in fruit extracts. Or you could say they had an activity of 6.6 and 2.5 AU respectively, whereby this is 6.6 and this is the 2.5 respectively. So this brings us to the end of question 2. Let's continue to question 3. Question 3, onion plants can be grown in a laboratory in a complete mineral iron solution. The solution contains all the mineral ions required for healthy growth. The diagram shows a young onion plant. So we have the roots, we have the bulb, and then the shoot. The effects of different strengths complete mineral iron solutions on the growth of onions was investigated. 3 concentrations of the solution were used. There is half strength, normal strength, and double strength. They want you to devise a procedure to provide a value comparison of the growth of shoots, bulbs, and roots of onion plants grown in these 3 solutions. Because we have 3 different solutions, half strength, normal strength, and double strength, we have to set up experiments of the 3 different solutions. So I say set up solutions of half strength, normal strength, and double strength. You have to use onions of the same species. They have to be the same age. And then you place the onions in these solutions for the specific period of time. However, their initial mass or length should have been measured. And then after the experiment, you need to measure the mass or the length of the onions and then calculate the change in length or change in mass of the shoots, or you could choose bulbs or roots. Then you can use repeats for each solution and obtain the mean. And then during the experimentation, because growth is enzyme controlled, you have to make sure factors that affect enzyme activity are going to be controlled. So you have to keep the temperature, the pH, as well as other factors like light intensity have to be kept constant. And then the experiment has to be carried out for the same period of time. Moving on. They say the graph shows the results of this investigation. So we see there is double strength, normal strength, and half strength. For the shoot, we can see half strength had a higher mass than normal strength, and double strength had the least. Actually, for all 3, double strength had the least, and half strength had the greatest mass. The shoot and the bulb of an onion plant are edible. The table shows the percentage of the total onion plant that is edible. When grown in half strength, we have to get the shoot plus the bulb because these are the only edible parts. Roots are not edible. So for the shoot, I came up with 4.4, which we have here. And then there is 0.6 in the bulb, which is about this one here. So adding these up is going to give me 5.0. And then the total will be that plus that plus that. So it's going to be 4.4 plus 0.6 plus this one here, which is 1.2. So the percentage should be what is edible divided by the total times 100, which gave me about 80.6% to 3 significant figures. And this was my answer here. Moving on. Here they say describe 3 conclusions that can be drawn from the results of this investigation. They want you to use the information in the graph and the table to support your answer. So when we go back to the graph, we can see half strength had the highest mass, but in the table we see the percentage of onion plants that is edible almost didn't change. 80.2, 80.6, and 79 are close. So here I said, as the strength of the solutions increase, growth of all onion parts decreases. And then the greatest effect of solution concentration was in shoots. Of course, we can see here, that is where we see the values are really high. So it was in shoots and the least was in roots. That is from the graph. Overall, the percentage of edible onion parts was not affected by the changes in solution concentration. This is from the table. We can see that the percentages did not change much. And down here they say, describe the information that is needed to decide if any of the differences seen are statistically significant. If you want to determine if they are statistically significant, then we have to use more plants. So repeat the experiment using more plants, calculate the mean mass, and then record your data, put error bars and see, and maybe use a specific statistical test and see if you can establish significance. So I said add more plants in each concentration of solution, and then calculate the mean mass and tabulate the results, including standard deviation. Draw a suitable graph and include error bars to observe any overlap, and then use a suitable statistical test to establish significance. Moving on. The table shows the mineral salts used to make a complete mineral iron solution. So we have the mineral salts, we have potassium nitrate, calcium nitrate, magnesium sulfate, and potassium hydrogen phosphate. So they say the effect of mineral iron deficiencies can be investigated by growing onion plants in solutions that have one mineral iron missing. This suggests how a complete mineral iron solution could be modified to show the effect of phosphate ions on the growth of onion plants. If you want to know the effect of phosphate, then it means it has to be removed. So we have to remove the potassium hydrogen phosphate, meaning carry out an experiment without potassium hydrogen phosphate. Now we have to find a way of replacing the potassium because in this case, our aim is to remove the phosphate. So you have to find a way of replacing the potassium. Maybe that could be by increasing the potassium nitrate because you cannot just buy potassium cation. You have to buy it as a compound. So you could replace the amount of potassium by increasing the potassium nitrate and then removing this completely. So I said experiment without phosphate by removing potassium hydrogen phosphate. However, this removal makes the solution deficient of some potassium, so compensate that by adding the removed potassium, probably using potassium nitrate. So here they say the pH can affect the uptake of mineral ions by plants. The chart shows the effect of pH on the uptake of some mineral ions. The height of the bar for each mineral ion indicates the uptake of that mineral ion. So we can see we have nitrate, phosphate, potassium, sulfate, calcium, as well as magnesium. So they say the optimum pH for the growth of these plants is between 6.5 and 8.0. They want you to explain why the optimum pH is within this range, and they want you to use the information in the chart to support your answer. If we look at this chart here, we can see they say the range is between 6.5 to about 8.0 because beyond that, we see the greatest concentration for each is about this range you can see. Beyond that, we can see this decreases if you go below that. And if you go above that, we see this also narrows down. So the range should be actually between this range here until 8.0, as you can see, because we will have the highest for each. Even this is going to be highest around that range. So I said, from the graph, the highest uptake of all these mineral ions is within this range. So if you use pH below 6.5 or pH above 8.5 for phosphate, you can see above this. If I take it back here, you can be able to see. For phosphate above this, we can see it narrows down there as well. So for pH below 6.5 and above 8.5, phosphate uptake goes down. The lack of enough phosphate will lead to less synthesis of nucleotides. There'll be less protein synthesis. There'll be less DNA replication. There'll be less mitosis. And ultimately, there'll be less growth. So the pH about 6.5 to 8 is optimum because below and above this range, the enzymes are denatured and growth can't occur properly. So this brings us to the end of question 3, as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.